Here we are instead of Semiamu. But say one good thing happened in spite of Semiamu closing. Elaine got to come from Germany to tell us about her research. Elaine's been connected with the OEH program, I guess it was when you started, since 2007. She's working on her PhD and undertaken research on a topic that's close to me. I've experienced allergic rhinitis for more years than I'm going to admit, and really looking forward to learning a bit more about it. And if we have time at the end of the presentation, I'll give you a case history just as another takeaway. Never mind. Elaine, you're on.
Thank you. So thanks everyone for coming to my presentation. So it's entitled Allergic Rhinitis, the Environment, Genetics, and How They May Interact. And so as a quick outline, I'll give a basic introduction of what is rhinitis for those who are not so familiar with the condition. And then I'll jump right into my PhD research. And I've chosen to present two aspects of this research. The first being focused on how climate affects rhinitis, and the second, how air pollution, genetics, and possibly how these two interact may affect allergic rhinitis. So as just a starter, rhinitis is basically inflammation of the nose. It can be characterized as nasal obstruction, itching, or sneezing. It can either be considered allergic, which involves also having itchy eyes, or non-allergic, in which case the itchy eyes are not there. You can also be also have rhinitis symptoms in association with exposures from your workplace, and then it's categorized as occupational. There's a strong link between rhinitis and asthma, and importantly, although rhinitis is not quite as researched as asthma, there is some significant negative impacts on the quality of life, and it also has some significant cost to school performance, work productivity, and your social life. So most of the studies that I was able to find on the economic evaluation of rhinitis, so how much it's actually costing, are US-based. So these numbers are in US dollars. And there's quite a variability or the range in what studies suggest that the cost may be, depending on whether or not they incorporate comorbidities or not, or if they include work performance or work performance or cost to social life. But it ranges somewhere between 2 to 5 billion US in 2003. And just an example here, the at-work productivity losses have been estimated between 2.4 and 4.6 billion in 2000, so it's probably a little bit higher now. But so this is a significant problem. And just to give you an idea of how prevalent it is, so this is a map taken from or produced by the ISAC study, and I'll talk about this study much more in detail later. But basically it's showing you the prevalence across the world of allergic rhinitis on this graph among 13 to 14-year-olds. And I don't really want to sort of draw any trends from this, but what I'd like you to see is that there's a lot of red stars, and that is indicating that the prevalence is actually 20% or higher. There's also quite a lot of yellow, those are diamonds, so that's between 10 and 20. So this is quite a prevalent condition across the world. And more importantly than sort of drawing trends on this is that there is this large spread of prevalence. So in some areas we have low prevalence, in other areas we have high prevalence, and we're not quite sure why that is. One of the hypotheses may be differences in environmental factors. What we do know is that there are risk factors to allergic rhinitis that have been previously identified. So we have the genetic risk factors, and it's well established that family history is important, ethnicity, and there's some research building on polymorphisms within the genetic DNA. And we'll touch on that later on in the presentation. On the other side, there are a lot of environmental factors that have also been identified. The most important is, of course, allergen exposure. There's also early life factors such as low birth weight or having a young maternal age of birth for the mother. Lifestyle factors, nutrition factors, and what I'm particularly interested is in the research looking at whether air pollution or climatic factors are also being associated with allergic rhinitis, whether those factors are playing a role in why this prevalence varies across the world. And yeah, most importantly, my research, the really novel part of my research is looking at how these genetic factors may be interacting with the environment to look at gene-environment interactions and how these may be affecting allergic rhinitis. So I just have one slide on climate change because it's a hot and important topic. And it's well known that the number and types of allergens are regional specific. So where you are in the world and what your climatic surroundings are do affect which allergens you're exposed to. And climate change is likely to affect the time, duration, and nature of the allergy season. And the graph that I have up here, I hope it's clear for everyone to see, is taken from Cisco 2011. And what this study looked at is basically how, in this case, temperature. What they have here on the y-axis is change in days to first frost. So how long do we have to wait before there was a first frost? And the graph on the top is change in frost-free days. So how many days there was no frost. And on the x-axis is the change in the length of the ragweed pollen season. And what they're showing here is essentially as it gets warmer or we have less frost, we have to wait for more frost or until the frost comes. We're not just pushing back the pollen season, but it's actually getting longer. And so symptoms, individuals who are suffering, they have to suffer for more months during the year. It's also possible that climate change will affect where these allergens are, so new allergens may be introduced. And there's also some research by the same author that showed that increasing CO2 was actually changing the level of allergens being released by particular plants, so it was going up. So there's some research that's starting to show that climate change may actually be increasing the length of the season, how allergenic each of the plants are, and so these are important consequences for people who are suffering from allergic rhinitis. And so this was probably the hardest slide for me to put together, sort of defining what is my PhD research. And I came up with 2 points. The first being that I'm interested in identifying whether environmental factors impact allergic rhinitis. And specifically, I'm interested in looking at climate and vegetation. And the second being air pollution. So that's sort of the first aspect that this is what I'll be mostly presenting on today. And the second component of my thesis is how does an individual's genetic makeup interact with their environment to affect their risk of allergic rhinitis and other allergic diseases, which I've sort of added on as we go on. And this will become a little bit clearer in the second half of my presentation. And it's been going on for probably, I don't know, 10, 15 years now, and they're right at the end of their phases, but it has led to over 500 publications. So it's a huge database, and I'm really grateful that they've given me the opportunity to work with it. What they've done in terms of rhinitis is establish that global variation in prevalence does exist. And so that comes with the map that I showed you. This wasn't known before, where there's been studies done here and studies done there, but it's always sort of differed in how they were done. Whereas now they've used a standard questionnaire, a standard method of assessment, and you can see that there is 4 reasons that are being investigated, this sort of global variation in prevalence. The second thing that they found is that the prevalences have increased over time. And so they repeated their survey about 10 years later, 8 to 10 years later, and they saw that most of the prevalences, especially in developing countries and especially in countries which initially had low and medium levels of prevalence, are now increasing. Countries which tended to have the higher level of prevalences in the baseline survey have not gone down but have essentially plateaued in time. And lastly, they have done many more, but what I'm interested in is that they've also looked at sort of how yearly reports of rhinitis were associated with climatic factors, and they found that for rhinitis, there was no association. And what I mean by yearly reports of rhinitis is that they used a question asking, in the last 12 months, have you had rhinitis symptoms? What the ISAC study has, but has not yet investigated, is that they have this data on monthly reports. So they've asked, did you in January have symptoms? Did you in February have symptoms? And so they have this very large database where you're able to look at an individual's yearly report of symptoms, and for in Canada, it would look something like symptoms go up in the spring and then go back down when the winter arrives. And so I'm interested in this, what about seasonality? Can we look at seasonality and how this may change with environment? If we think of a place like Canada, you're much more likely to get this seasonality effect, which may be either more associated with environmental factors. So maybe that's something that we should be looking at in terms of these climatic variables. So the first thing I found out is that global studies are challenging. I guess the easiest way to say that is that what spring is in Canada is not exactly the same thing, or it's not at the same time as spring in Brazil or in Australia or something like that. And so it became quite evident that I was going to have to work around these problems. So in previous studies, rhinitis symptoms are typically classified as either seasonal or lasting all year round. However, there are issues with this classification, apart from the global studies. Firstly, molds and pollens are not always seasonal. In some areas of the world, They're there all year round. Yearly symptoms may not be present all year round. Since individuals are sensitized to both yearly and seasonal allergens, and symptoms do not necessarily occur in strict conjunction with the allergy season. And I'd also like to add the point that I mentioned before for global studies, is that seasons differ in different places of the world. And so I was able to, very quickly I realized that it was going to be difficult for me to sort of identify what is seasonal allergies and apply that to the entire world. So I started reading, and thankfully I found this new rhinitis classification, which has been proposed by the Allergic Rhinitis and Impact on Asthma Working Group. And they've recognized these problems, and they've come up with two different classifications for rhinitis, which are much more easily applicable on a global scale. So we have intermittent rhinitis, which means that the child or the individual has reported at least once, but never for 2 consecutive months. Persistent rhinitis is when a child or an individual reports rhinitis for at least 2 consecutive months in a row. So they have to say January, February, or November, December, 2 months in a row. And there are certainly more detailed definitions I'd like to mention, which include number of days and stuff like that. But for when studies only have monthly data available, these are how the classifications are being used. Before we go any further, I'd like to emphasize that these are not interchangeable with the past definitions. So intermittent rhinitis does not mean seasonal rhinitis. However, it does better describe the patient's experiences, these new classifications. And importantly, it doesn't necessarily stem from the allergic origin. And this becomes important in the global studies. So I'm able to take these definitions and apply them everywhere in the world. And intermittent rhinitis in Canada is the same as intermittent rhinitis in Brazil. So then I was ready to start, now that I had sort of identified the problems and worked through the initial difficulties. So I'll be presenting some results from a global study which examined the role of climate on the exacerbation of intermittent and persistent rhinitis symptoms. And this is a work in progress, so I'm still sort of tweaking the final analysis, but we are happy to present what we do have already. And we've identified 2 research questions. The first being, are there global variations in the prevalence of intermittent or persistent allergic rhinitis? We're interested in sort of creating a similar map as before to see whether this variation exists. Are there particular areas in the world that are reporting higher levels of persistent symptoms? And so we needed first to look at whether global variation also exists in when we classify in these 2 different methods. The second, which is sort of the more novel approach and what I'm most interested in, is to determine whether these intermittent or persistent symptoms are associated with climatic factors, and we have information on temperature, precipitation, or water vapor pressure, or whether they're also associated with vegetation cover. And at the center level will become clearer as I work through the methodology. So this is probably difficult to read, but I just wanted to give you sort of an idea of where the data is coming from. So the study population, it's the ISAC phase 3 data, so it just means that this data was collected between 2003 and 2005 as part of the phase 3 of ISAC. And you can see that the numbers are quite large, so it's about 200,000 children aged 6 to 7 and 300,000 from the older age group, 13 and 14 years old. And what we end up doing is we take all of these children and we group them into their centers. So within a country, we'll have centers, for example, in Vancouver and Toronto, and we've managed to group them into the centers, as has been previously done. And we're up with about 132 centers for the younger age group and 222 centers for the older children. And so these are the numbers that go into the analysis. So it's a center level analysis and not at the individual level. So I have one slide on methods for those interested in statistics and to sort of understand how we've done this. So my outcome is, again, the center prevalence of intermittent and persistent rhinitis symptoms. So not at the child level, but sort of in Vancouver, 20% of children have rhinitis. That's my outcome. My predictors are temperature, precipitation, vapor pressure, and vegetation. And so the 3 climatic ones, temperature, precipitation, and vapor pressure, the data was obtained from the International Panel of Climate Change, and I've sort of given you an example here of what it could look like with temperature. And each of these lines, the different colored lines, is a center, and I've grouped them into their climatic zones just to help give a trend of how the differences may arise. So the warm, temperate, dry winter is sort of Europe, or something like Canada, compared to the equatorial data. So you can see that there's quite a difference when you're looking at sort of how climate may affect allergic rhinitis. There's a difference between these areas You can see here in the blue and snow, this is the mountains in Chile. And so you have the opposite trend of what you would have for the rest of the snow areas. And so what we take this monthly data, we gotta go back, yeah. And we take the mean, I look at the max, I look at the minimum of each of these monthly estimates. So the mean monthly estimate, the maximum monthly estimate, as well as the difference and the standard deviation of the monthly estimates. And the reason I look at these last two, the difference being between the maximum and the minimum, so the maximal difference in the monthly estimates, and the standard deviation, is that it gives us a measure of the variability of the climate in that place. So in Canada, it gets really hot and it gets really cold. And so your difference or your standard deviation would be much larger than in a center, for example, on the equator, where they don't get this sort of like variability there. And so that's what the last two exposures are catching. And I have each of these 5 for temperature, precipitation, vapor pressure. And the vegetation data is actually quite novel and new, and we're pretty excited about it. I'd like to thank Sarah for helping us link it, take it from whatever the database was and link it to our different centers. But my understanding is that they've used satellites to take pictures of the vegetation across the world on 16 days, on a 16-day basis, and we can see how these differ across the world and how they differ over time. So we also have these monthly vegetation data, and I also obtain the mean, the max, the min, and so on. For the statistical analysis, I'm using a linear regression mixed model. I include country as a random intercept. My fixed effects for each of the main exposures, so up here the predictors, are fixed effects. And I additionally adjust the models for sex, population density. When I'm looking at climate, I adjust for vegetation and vice versa. And these variables are all at the center and the country level representation. And I also adjust for an economic variable, so the gross national income per capita. So that's a lot of methods, so we can now look at some results. So these graphs show the prevalences, so just a scatter plot of the prevalences. On your left is intermittent, and on the right, B, is persistent rhinitis symptoms per age group. So on the x-axis, we have the younger children, 6 to 7, and on the y-axis, we have the older children. And all I want to show with this graph is that there is a very strong correlation. It's actually not on here, but for both symptoms, it's 0.78. So if a center has high prevalence in the older age group, they're also going to have a high prevalence in the younger age group. So from here on in, I'm only going to present the results for the older age group, just because it's less numbers and it's easier to explain and for everyone to understand. And so for the 13 to 14-year-olds, this table shows the range and percentiles of the center prevalence of both intermittent and persistent rhinitis symptoms. And there's a little star there that goes at the bottom of the graph, which indicates, or which tells you that this is not representative of world prevalences. And what I would like to emphasize with that is that these samples, these centers were not randomly selected throughout the world. I mean, there was a selection process, and so we can't just say, okay, this is representative of the world prevalence. But we do have 233 centers, and so it is a good, it's the largest collaboration on this, of this size. And what we see is that for both intermittent and persistent symptoms, we have a very large range of prevalences. So there are some centers that say 0%, none of our children are suffering from intermittent symptoms. And on the other side, we have centers which are saying at the maximum level, 50% of children are suffering from intermittent symptoms. And so we still have this very large range that I think it's actually even larger than when we only looked at allergic rhinitis on a yearly basis. And so to answer the first question, we do get this very large range in prevalences across the world, and we're not quite sure, again, why. This may be sort of environmental factors, and that's what we're interested in finding out. And I'm always interested in knowing, well, where's the highest place? So I threw down the numbers here, that the intermittent rates are highest in the Congo, and lowest in Brazil and the Kingdom of Tonga. Persistent rates are high in other places of the world, Thailand being the highest, and the lowest is in the Gabon and Nigeria. And so when I have a map, and we didn't throw it up here because it becomes too much data for people to see, and it's difficult to obtain a sort of idea of where the rates are, but what we have seen, one of the consistent trends that we've seen, is that Europeans tend to suffer more frequently from persistent rhinitis symptoms than intermittent symptoms. And we're not actually sure why, so I'm still sort of tweaking that and seeing if I can identify other trends, or whether there's just sort of this variation that we can't explain, or that we're looking to explain. So this was probably the most complicated slide I'm going to show. So we'll go through it slowly. And I'll start with saying that this is the results for between country associations. And so what I'm doing here in the model is looking at associations that differ between like Canada and Germany and Brazil, and not between the actual centers. So it's not a Vancouver-Toronto analysis, but really Canada and Germany. And so between country associations for intermittent and persistent rhinitis symptoms with all of the environmental factors. So it may be hard to see here, but temperature is the first one on the red dots, precipitation is in blue, vapor pressure is in green, and the vegetation is brown on the end. And for each of these factors, so for each of temperature, I have the mean, the max, the min, the maximal difference, and the standard deviation in that order. So if we just start with intermittent symptoms and temperature, right here on the red on the first dots, you can see that the mean, the max, and the min are significantly elevated. So there's a positive change in prevalence per 100 children per increase in IQR of exposure. So what that translates to is places with higher temperature tend to have higher prevalences. And interestingly, when you look at the two other measures, the difference between the max, the min, and the standard deviation, you have sort of the opposite trend. So places like Canada, which have this really high variability, actually seem to have a decreased change in prevalence, so it's protective compared to the others. We have a similar trend for the vapor pressure in green, although the last one is not statistically significant. That's not necessarily surprising, as vapor pressure and temperature tend to be very highly correlated. And for precipitation, all of the estimates tend to be above. So if it rains more, the change in prevalence per 100 children is elevated. As well as the measures of variability, so places like India, where you get a lot of rain in one month and then not a lot of rain in the other. This also seems to sort of increase the prevalence, which is the opposite to precipitation and vapor pressure. And for vegetation and intermittent symptoms, we don't really see any consistent trends. When we look at persistent symptoms, which is the graph on the right, the first thing we see is that we don't get these strong associations with climate, or these significant associations with climate. So there does seem to be somewhat of a difference of how the climatic factors are actually influencing the different types of rhinitis symptoms. However, when we look at the vegetation estimates on the last series of dots there, although the last two are not statistically significant, or the second one, the first three are definitely elevated. So areas which have more vegetation appear to increase the prevalence in persistent symptoms. Places with more vegetation. They're separate outcomes. So I'll just repeat the question. Kay is asking whether they're all separate models. And these results, so the vegetation is adjusted for the mean of the climatic ones, and the climatic ones are adjusted for vegetation. And I have done quite a lot of modeling where I've put the precipitation and the temperature and the this and the that, and so we've done a lot of that testing, and we always get these sort of significant results. And also what I'm not showing here in this presentation is that we've adjusted for like climatic zone. When I've showed that graph where they were split into the different categories, snow, equatorial, we also adjusted for that, and we still see this, at least for the intermittent symptoms, they're very consistent, that we always find these elevated effects no matter what goes into the model. So interpretation, I like to emphasize here, current results, is that there is substantial variation in the prevalence of intermittent and persistent rhinitis. So we still see this difference across the world. And sort of a one trend that we may draw is that Europeans suffer more frequently from persistent symptoms than intermittent symptoms. But we don't really see so it's not as in only in South America do they have very persistent symptoms. We don't see that sort of concrete trend. When we look at the associations with environmental factors, there's significant associations between the center prevalence of intermittent symptoms and several of the measures of climatic variables that we looked at. For vegetation, I say here not consistently relevant, But I'd probably add, if I could read you this slide, is that we do see, at least for the mean and the maximum, an elevated trend for the persistent symptoms, although nothing for intermittent. So now I'll start the second component of my presentation. And this was sort of this one is finished, so that's good. It's on air pollution, genetics, and gene-environment interactions. And so just as a quick background slide, I'd like to introduce what we know about air pollution and allergic rhinitis. So ambient air pollution results are conflicting. When we compare cities, it's quite conflicting results for allergic rhinitis. And it's possible that traffic-related air pollution may be more relevant. And the reasons for this is that we get more we're capturing sort of source mixtures when we estimate these things. The individuals may be more exposed because they're closer to the sources, but importantly, we're able to capture within city variability. So individuals living on Broadway probably have a very different exposure to some of the more, trying to think somewhere with no traffic in Vancouver, Stanley Park, somewhere near Stanley Park. So the results for traffic-related air pollution may be more relevant, especially for asthma, where the results are quite consistent. So we also want to take this approach and examine allergic rhinitis. And I've taken this little diagram from a paper from Saxon, as well as David Sanchez, who I believe was here in November or December last year to give a talk. They had a great paper explaining how air pollution could cause allergic diseases. And I won't try and explain this into too much detail, but what's important to note is that these are diesel particles, the little black ones coming in. And the idea is that they interact with your airway epithelium or your mucosal lining, and they lead to a variety of immune effects, but importantly, it causes the increase in the creation of reactive oxidative stress, or ROS, which, if left untreated, can lead to inflammation. And so the idea is that the body, when this production of ROS occurs, these oxidative species, should be able to metabolize the foreign bodies as well as handle the oxidative stress. But when this doesn't happen, it can lead to inflammation as well as the development of allergic diseases. And so individuals who have a diminished ability to metabolize this oxidative stress may be at increased risk of air pollution. And this, you could think of these people as sort of genetically susceptible to the effects of air pollution. And here's a much simpler figure, which I like to sort of explain how gene-environment interactions may occur. And there's definitely a variety of possibilities, but this is a simple one for me to explain. So if you can imagine that environment is air pollution, so as air pollution goes up, individuals who are homozygous major for whatever genetic SNP you may be looking at, so they have two of the same alleles, have no risk. However, if you have a major allele and a minor allele and are heterozygous, then you may have a more increased risk. And if you're a carrier of two minor alleles, you may have an even more increased risk. And so this is how maybe genetics may interact with the environment, where these individuals the homozygous minor allele will be genetically susceptible to air pollution, whereas the homozygous major allele carriers would show no association. And a real example is the story behind CD14 asthma and endotoxins. And you can see here the three different genotypes, so CC, CT, and TT. And depending on the environment, so either low endotoxin, which is the lower line, individuals with a TT allele would actually have a decreased risk for asthma, whereas individuals with the same allele but who are exposed to high endotoxin would have a higher risk for asthma. So this is sort of a real-life example of that gene environment put into play. So this leads me to the Traffic Asthma and Genetics, or TAG project, which is led by Chris Carlston and Michael Brower. And the initial project, the project generally aims to assess whether exposure to traffic-related air pollution interacts with the child's genetic profile to impact the risk of developing childhood allergic diseases. So we're interested in looking at gene-environment interactions in the context of allergic diseases and traffic-related air pollution. And what I find is really cool about this project is that they've managed, through their collaborations and networks, to sort of obtain data from 6 different birth cohort studies, 2 of which are from Canada, the CAPS and the SAGE study, and 4 others from Europe. And they've been able to combine all of this data into a central database. And what's really unique about these birth cohort studies is that they have individual level NO2 exposures that they both address. So it's not just that everyone in Vancouver gets the same NO2 level, but really at the birth address they have sort of an NO2 estimate. And these were derived using either land use regression models, which I think we've had a presentation on before, as well as dispersion models for one of the cohorts. Importantly, they also had similar outcome and covariate definitions across the cohorts. And they had all taken an interest in genetics. And so they had available information on some single nucleotide polymorphisms, or SNPs. And this is sort of the difference between that I showed before if you're a CC or a TT. So these are the SNPs here in different oxidative stress, inflammation, and immune system development genes. And the initial goal was in the oxidative stress genes because of this sort of diagram that I showed before and the hypothesized mechanism by which air pollution may actually lead to inflammation. But there is increasing evidence that inflammatory and immune system development genes may also be important. So this is the study population for this project. You have the 6 birth cohorts. Caps and Sage are from Canada, Brahms is from Sweden, Gina and Lisa are the German birth cohorts that I work a lot with, and Piama is from the Netherlands. What's important to note is that they were all approximately started in the same year, so the children had similar birth years and are approximately the same age. They're all birth cohorts. They were recruited either during pregnancy or at the time of birth. And also what's important to note is that they're not all population birth cohorts. And so these cohorts were initially designed, for example, the Sage and the Bomza cohort are nested case control cohorts. 3 of the cohorts, CAPS, Genie, and Piama, have interventions built into them. And CAPS is actually a high-risk cohort, so to be included, one first-degree relative had to have asthma, or 2 first-degree relatives had to have some other allergic-mediated disease. And so we were pretty concerned, not concerned, but aware of this, as when we pooled all the data, we wanted to pool it in order to obtain maximal statistical power to test the gene-environment interaction. So we did a lot of sensitivity analysis where we examined the impact of including each of these cohorts. And this will become clear in the results. So a method slide, the outcomes that I was particularly interested was, of course, allergic rhinitis, and this was defined at either 7 or 8 years, as well as allergen sensitization. My predictors for this model, again, I'm focusing on air pollution now. So I had NO2 for all of the cohorts, and this was assessed individually at the birth address. And for the majority of the cohorts, we also had PM2.5 mass, PM2.5 absorbance, and ozone estimates, individually assessed again. And here I'm going to present results on 8 SNPs related to oxidative stress. And for those of you who are familiar, it's the GSTP1 gene that we focused on, inflammation being the TNF gene, and immune system development, and the focus was on the TLRs, or toll-like receptors. The statistical analysis that I followed was to use a logistic regression, assuming a dominant genetic effect. And what this last part suggests is that if a child had 1 or 2 minor alleles, we considered them minor alleles, and we compared them to the major allele group. The graph that I showed had this sort of additive effect, and we would have combined the 2 lines, the 2 top lines together, and compared it against the bottom line. So the results that I'll be showing are presented pooled and by cohort, and the reason that we do this is that we get this sort of pooled effect, but we also wanted to ensure that it was consistent across the cohorts. And so by presenting it pooled and by cohort, you can see whether it's approximately the same or whether the cohorts are really all over the place. And all the models were adjusted for study, city or region, sex, intervention when it was applicable, maternal age at birth, family history, and environmental tobacco smoke. So here's just a graph of the NO2 distribution by area. It's probably a bit small on the x-axis, but the Netherlands is in purple, Sweden's in red, Canada is in green, and you can see the difference there between Vancouver, which is high, and Winnipeg, and Germany is in orange. And the 2 points I'd like to make on this slide is first, we have a good overlap between the cohorts. So we're not comparing areas with extremely low and extremely high and having to extrapolate between them, but we actually have good overlap between. The second point I'd like to make is that the range of exposure that we're looking at is greater than if we had any single study alone. And so we're able to compare like low levels all the way up to very high levels and see how the association changes rather than any one of them which would have had a smaller range on their own. So here are the first results. So this is looking no genetics whatsoever. This is only looking at allergic rhinitis and the association with air pollution. On the left is NO2, and on the right is PM2.5 mass. And it's a little bit hard to see. My dots came out fuzzy. But the pooled is the first association on both graphs, and then the cohorts follow. And what we see for the pooled NO2 and PM2.5 mass is that they were elevated, and it was statistically significant for PM2.5 mass. However, for PM2.5 mass, when we removed the Piama cohort, and that's the last cohort on the slide there you see is very elevated, our pooled result for that pollutant dropped almost to null. And so although we do report this pooled estimate, we also we emphasize that this is primarily due to only one of the cohorts. When you remove Piama, the rest of them are very much null. And so we conservatively say that we didn't find consistent evidence that it's mostly attributable to this one cohort in the Netherlands. So now for yet environment, and this is just allergic rhinitis and genetics and SNPs. And I only present the significant results, but there are also 6 other SNPs that we tested that were not significant. So the RS 1-800-629 is the TNF SNP for inflammation, and the other SNP, the 192, is in the TLR4, intron region of the TLR4 gene. And what we found for these two is that the pooled result was elevated, so minor allele carriers who carry at least one minor allele will have an increased risk of allergic rhinitis compared to the children who had 2 major alleles. And this was consistent no matter which of the cohorts we pulled out. So we were able to take out any of the cohorts, and the confidence intervals occasionally crossed the significance line, but the estimate always remained identical. Not identical, but very, very similar. So it wasn't as though one cohort was entirely driving the association. And we also adjusted, as I said before, the studies were, some of them were case controls, some of them were high-risk cohorts, so we also did quite a variety of adjustments for cases, removing cases, and always our result was consistent, so we're quite confident on this result. But the real purpose of this was not to look at air pollution effects and not to look at genetics, but really to see if the air pollution effects differed by genotype. So what I have here is only the SNPs, these are numbers. The first 2 are from the GSTP1. The 1-800 is the TNF, which is inflammation, and the rest are from immune system development genes, the toll-like receptors. The numbers here in the percentage are for each of the SNP in the pooled population, the proportion of which were considered minor allele carriers. So they had at least one minor allele compared to the children who had 2 major alleles. And what we find is that there isn't really a difference in the association. Well, first of all, none of the associations are really significant. And there didn't seem to be any difference of the association between the NO2 between the 2 genotypes. So it's not that, for example, the children that were minor allele carriers in purple had a significant risk of NO2 compared to the others. We also ran a model. This is a stratified approach. We also ran models which included a sort of an interaction term between genetics and air pollution to formally test the interaction between gene-environment associations, and we found no significant results for allergic rhinitis. So the next 2 slides are actually taken from Elaine McIntyre's work, who was a postdoc here also working on the TAG project, and she did a similar analysis but was looking at asthma at 7 to 8 years. And so here are her stratified results, and you can see that for her pooled result, the red box, the minor allele carriers, are actually at a significantly increased risk. So the association between NO2 and asthma is significantly elevated, but only among this genetically susceptible population, these minor allele carriers. And the pooled result is significant, and a lot of them are actually off the chart because the sample sizes are small when you start looking per cohort, especially for the smaller cohorts. But you see that the red tends to be above the blue in all the cohorts. And when we pool this, you get a significant result. And so that's the beauty, really, of TAG, is that you take advantage of all of this data, you pool it together in order to be able to obtain the statistical power required to look at something like this. So this was for one of the genes, one of the SNPs in the GSTP1 gene, which is involved in the oxidative stress pathway, so completely in line with what was hypothesized at the beginning. She also looked at the other SNP within the same GSTP1 gene involved in oxidative stress, and again found a similar result where the minor allele carriers seem to be at an increased risk of the effects of NO2 on asthma. So when we summarize what so far has been done for the TAG project, for allergic rhinitis, we found no evidence of gene-air pollution interactions. We found no consistent evidence for main air pollution effects. And I say that again to say that although the PM2.5 result was elevated, it was associated really with this only the Piama cohort showed that effect. However, children with minor, this particular allele in the TNF SNP or minor 192 allele in the TLR form may be at a higher risk. And what's interesting about this genetic result, first of all, the 1-800-629 allele has been previously associated with allergic rhinitis in three studies looked at it. 2 of them found positive results. But the studies were about 200, 250 children. So we're the first to really show it in a substantially larger population. And this allele, this particular SNP is located in the promoter region for that gene. And so when there is this SNP, when it's not there, it appears that more of the TNF protein is being produced, and that protein has been found to be higher in individuals with allergic rhinitis. So there is sort of a potential biological mechanism behind it. For the TLR4 SNP, we're the first to find it associated with allergic rhinitis. It's in the intron region of the gene coding for that protein, and so there isn't really a lot known about it. So we're hoping that others will take a look at this and try and replicate in other populations. We also did the same thing for sensitization. I didn't show anything because we didn't find any significant results for that particular outcome. It is important to note that the sample size was almost half because they did RAS tests or immunological data, and so it's not all the cohorts did it for every child. For asthma and wheeze, which again is being led by Elaine McIntyre, she found that children with minor GSTP1 alleles, so 2 SNPs in the same gene involved in oxidative stress, appear to be a susceptible population to air pollution. So I'd like a few acknowledgments. My co-supervisors, Drs. Chris Carlson and Michael Brower. My committee members, Drs. Andrew Sanford, who's also part of SPPH, and Joachim Heinrich, who I work with a lot in Germany. Allergen NCE for funding the TAG project and for providing personal support. CIHR, TAG collaborators, and I don't really want to gloss over this because there's been quite a lot of effort to sort of obtain all this data from the 6 birth cohorts and pool them all together. There's been a lot of meetings and a lot of collaboration, so that's a lot of work on the TAG collaborators, as well as the ISAC collaborators for allowing me to use their huge data set for my doctoral research, and again, Sarah Henderson for helping to link the vegetation from the satellites. So I'm only in town until Sunday, so I'm here all afternoon if anyone has any good ideas or wants to talk, or else the best way to reach me is by email. Thank you.
Great. Do we have questions in the room here? No?
It's kind of a big picture question. I don't know if this is working, but it's going to be helpful, I think, for me and others, and certainly you, when you think about the ISAC data. It's interesting, this idea that what is the trend, which ISAC, I think, is really well suited to demonstrate, as you said. And it kind of seems, the data is extremely complicated, but it seems that the rhinitis, the allergic rhinitis trend might be a little bit stronger in terms of increasing in the last so many years, say decade, than asthma. There's a lot of caveats there, but in general, I think they would say, as far as I can tell, the increase globally, with all the caveats, is greater for rhinitis than asthma. Right?
Yeah, but also in general, the prevalences in the end, so the allergic rhinitis is more common than asthma. And so the change in the increase may also...
So you think it's just a relative thing? Because anyway, if it really is true, I'm just kind of wondering, since you've been so involved, whether people sort of in that real nitty-gritty community are sort of hypothesizing on why that could possibly be, if there really is a difference in the trend between the two conditions.
I can tell you that they followed similar trends, so asthma and allergic rhinitis went up and they both went up in countries where it was low beforehand. These tended to be the low-income countries, or low developing countries. In areas that were developed, they generally plateaued for both of the disease, but whether they've looked at sort of the relative increase, I don't know. No, I don't know.
Thanks so much. It was really fascinating to find out what you've been up to, and you presented that so clearly, so really appreciate it. I have a couple of questions. One, I think you said at the beginning that the prevalence of perennial rhinitis was higher in the European countries than in the developed countries. Is that right?
No. So I also looked at, my hypothesis was that persistent would be more common than intermittent. And I was quite surprised to see that the majority of countries have higher intermittent rates than persistent rates. And a stark difference is really in Europe. So I took a ratio of the two. And in Europe, it looks like they suffer from persistent rhinitis symptoms much more frequently than they do intermittent symptoms. And everywhere else seems to be the flip.
Okay, so what I was wondering was whether or not that might be because European compared to a lot of the developing countries, people spend more time indoors, and it may be more likely to dust mites and things like that, and if there'd be any discussion of that. And then my second question is, you put country as a random effect in your models, and I wondered why, and I wondered if that might be overmatching on some of these climatic variables that you were using.
Okay, I'll try the first one because I think it's easier. So you were asking whether maybe Europeans are spending more time indoors and that's why they're suffering from persistent symptoms. I mean, it's definitely, again, it's not, this persistent does not necessarily mean all year, because they can only say January and February, and then they would be classified as persistent, whereas because they have 2 months in a row. Whereas an intermittent sufferer could say it 5 times, but never in a row. And so, yeah, it's like there are limitations to the definition, but it's sort of as good as we could have done, because the seasonal definition is really challenging to try and say when is the growing season in this country, when is the growing season in that country, and to do it for 100 countries was making a lot of assumptions. Persistent is typically considered the more severe type, and it is possible. What I haven't done yet is sort of to compare Europe to other similar, I would say, countries, maybe like Canada or the States, where I would say that we have equal time indoors here as we do there. And the second question. Yeah. I don't have a good answer. I work closely with the group in London who did sort of the air pollution, Ross Anderson's group, and Barbara Britland, who does the statistics. And I wouldn't be able to tell you, we included it because we wanted to allow it to vary by country. So when we read it, they have a paper out where they didn't include random intercept, and the association went one way. And when they included the random intercept or looked at it per different countries, then you had these varying different associations. And so that is why we put it in. But it would certainly be interesting just to take it out and see. The initial idea was to report by center and actually just account for country and report the results by center. But then we get in a problem of sort of clustering effects because we're comparing Vancouver to Toronto in Canada. But we're comparing like Munich to Dresden, which is a city that's very close. And these are considered equal centers. And so we thought that looking at it by country would be a better way to sort of account for the differences.
I'll just say what I thought you might say, but you didn't.
Okay.
And that was, I thought you were going to say that you had some concern that that the way people report these symptoms might vary by country by some systematic cultural difference or something, but that wasn't part of the discussion?
They've actually looked at that in a separate paper, and what is not on here, but what will be done is to look at English speaking versus non-English speaking. And that seems to be a larger confounder, because the questionnaire in English is the same in English-speaking countries. And also in countries where literacy was a problem, where they actually gave the questionnaire by speaking rather than actually by answering. And so not so much between the countries, but really between these different literacy levels or languages.
I got a question. What does ISAC stand for?
The International Study of Asthma and Allergies.
Something else.
C.
Okay.
Consortium. Thank you.
Thank you. Other questions? Sorry? No. Not getting any signal from here.
All right, well, it's great to see what you're up to.
What I've been doing.
I was just wondering, for the countries that have multiple sites, what was the variation within country? And is it comparable to what you saw between countries? So if you look at like Vancouver and Toronto type of thing.
So one of the big problems with that, with the within country, is that we have countries like Brazil that have 13 centers. And then we have 2 countries that have 6 or 7 centers, and then everybody else has 2 or 3. And so, I mean, obviously the variability was huge in countries like Brazil, but in the Netherlands, who have something like 4 centers, like how big is the Netherlands? They're quite, there's no variability, and so we had a lot of problem teasing that out. We did run the associations and pull center out to see what the effects were, and they're essentially null across the board, and that's not super surprising because we also have to account for country. So the differences between country are much larger. We also have much more power, as only about 57 of the countries have more than 1 center. So we have to kick a lot of the people, or a lot of the other countries out.
I had persistent rhinitis for a number of years, back in the early 80s. And it was, yeah, it got to the point where my wife wasn't getting enough sleep because I was blowing my nose all night. And that got me sent to an allergist where I finally realized, oh, okay, it's allergens that are causing my nose to run. And he gave me some really good advice on how to reduce my exposure to allergens. The first was to eliminate hosts for dust mites in my house and clean up dust and so forth. And I actually listened to this doctor, which is really quite an unusual situation for me. But anyway, I did a lot of what he told me to do. I observed when I went up to my cabin in the spring and ran a lawnmower over tall weeds, that my nose would start to run and would run like crazy for several days. Then I tried wearing a respirator, a dual cartridge HEPA-filter respirator, like your N95 or whatever. No symptoms. Voila, right? What's triggering my rhinitis is very small particles, and you can filter those out of the air you're breathing. Heck of a good move. When you do certain jobs in your house that disturb dust, try wearing a respirator. You know, it'll stop your nose from running.